Hey guys, welcome to today's video. It is on replication of DNA. So the learning goal for today is to look at the key players in this process of replication. First being helicase, the second being DNA polymerase, and we are also going to make reference to the direction of replication. So this is taken from 7.4 of the text, and as you can see, there's a whole bunch of key terms down the side here. I'm not going to directly read those, but we'll make reference to them in this video. So DNA replication is the process by which DNA makes a direct copy of itself. But why is this important? Well, here is an image of an egg and a sperm cell penetrating that egg in this process of fertilization. What becomes of this is what's called a zygote, and that zygote will divide by mitosis to eventually create an embryo, which will develop into a fetus, which will further develop into a little human, which will have over trillions of cells. And it is important that every single one of those cells has the exact same DNA as the very original cell. So we need to make sure that this occurs. And that is the process of replication of DNA. So this process occurs in what's called the interphase stage of the cell cycle. And when it comes to the replication of DNA, there were three theories. The first is the semi-conservative. This was believed by Watson and Crick, two scientists who made massive contributions to our understanding of DNA. The next is conservative, and then we have dispersive. Semi-conservative says that DNA, double-stranded, splits into two single strands, and then direct copies are made of each of those single strands. The conservative says that kind of this photocopying process takes place, whereas dispersive says a bit of both. This chunk makes a copy over here, but this chunk makes a copy over there. And it wasn't until 1958 when the Messels and Stahl experiment proved that it is actually the semi-conservative process that takes place. So before we talk about the process of replication, let's talk about the structure of DNA to recap last week. We have in DNA a deoxyribose sugar group, which is attached to a nitrogenous base, and this sugar is also attached to what's called a phosphate group. The sugar phosphate backbone is made up of a sugar attached to a phosphate, attached to a sugar, attached to a phosphate. We have nitrogenous bases that bind to that complementary pair, which just means whenever you have an A, you have a T, whenever you have a G, you have a C, through hydrogen bonds, but over this side we have covalent bonds, which are much stronger because you don't want your DNA falling apart. You can also see that there are labels to this sugar phosphate backbone. We have what's called the 5' end and the 3' end. The backbone of DNA is parallel because they face each other, but they are actually what's called anti-parallel, and it means that though they are facing each other, they're running in opposite directions. The way in which they are running in opposite directions is chemically. And it comes back to this sugar. You can read a little more about that in the textbook. This idea of the backbone DNA running in opposite directions, anti-parallel, is important in the process of replication, as we are about to soon see. So there are many enzymes involved in this process. But for the sake of understanding what's going on, this diagram does a pretty good job, even though it's a little bit more simplified, but just keep that in mind. So the first step, the first thing to point out before the first step though, is that DNA, as you can see, is a double helix, and that helix that is kind of coiled up is firstly straightened out. The main enzyme, one of two main enzymes that we'll now point out, is helicase, and helicase is involved in It's called the unzipping, and it just splits those two strands of DNA side by side. This part where helicase unzips DNA by their nitrogenous bases is called the replication fork. So single strand proteins then bind to this now separated DNA strand to hold it together. The next main enzyme involved in replication is DNA polymerase. And DNA polymerase races along and adds nucleotides to the original strand, creating a direct copy. As you can see down here, this is called the leading strand, and this process happens silky smooth because DNA polymerase can only add new nucleotides to the 3' end. This happens easily on the leading strand. If we follow this, we have this strand here. It's the 5' end, which means the opposite end of that 5' end would be the 3' end. And so if helicase is unzipping the DNA, then polymerase can continue to race along and add new nucleotides really easily. This is not the case on the opposite side where we have the lagging strand, but we have this elegant method that takes place with a few more steps involved, and because of that it often lags behind, so that is why it is called the lagging strand. We cannot add, DNA polymerase cannot add new nucleotides to the 5' end. And if you look here on this strand, 3', this is the one that's been constructed, so therefore this would be the 5' end. It cannot happen, so we have a few more steps involved that make this process available. So the first step is DNA primase comes in. It's another enzyme that enters the game here. And what it does, it adds what are called RNA primers. And it's these little sections of RNA that essentially tell polymerase, hey, start over here. Now, if you've ever done any sewing or stitching, which I have not, this can be thought of as back stitching. So you're still going in this direction, but you're kind of working back and then jumping forward as you go. So that's what's happening with DNA primase and DNA polymerase. So this RNA section is added here, then DNA polymerase jumps over and then continues to add DNA here. There's a problem where if we have all these little sections forming, RNA bumps into DNA. So we have another protein, enzyme, called DNA ligase. And DNA ligase essentially takes off those little starters, those RNA primers, and replaces them with DNA and fixes up all the gaps so it makes it nice and smooth. These sections that are formed through polymerase working in parts are called Okazaki fragments, and there's another interesting story there which you can check out in your own time. There is an excellent computer animation showing this process taking place that I have added the link to in the description of this video, so check that out after this. This process of replication occurs in eukaryotes, such as animal cells, humans, though it is different for prokaryotes. They replicate DNA and split their cells through a process called binary fission. Like we said last week, the DNA in our cells is so long. So there is another image that I'm going to talk to before we move on. Now, if you look at this, we have two strands, that's our DNA. If we had helicase working here and splitting that DNA and just working in one direction, remember how long that DNA is in every single cell? It would take a long time. So there's a couple more little processes just to be aware of here. Notice that there is an arrow pointing this direction and an arrow pointing this direction. So this whole process is happening at multiple points along the DNA. Helicase is going this way, going that way. Then we have it happening here again. There are multiple replication forks. Because of this happening in multiple places, it creates what's called a bubble. Now this bubble will eventually meet up with another bubble, and then we will eventually have what are called two daughter DNA molecules, and it was taking one double helix of DNA and creating two double helix DNAs, and that is the whole goal, whole purpose of replication of DNA, because every single cell needs to have its own set of data. And that is all for today. So let's get on to some questions. This refers to what we covered last week, or in the readings at least, and it is from QCAA, and it says define the term gene. So pause the video here, 1 mark, so 1 definition, 1 point. This is the response, and it's quite simple. It says regions of DNA that are made up of nucleotides. You might have different alternatives of the same answer. The textbook says something similar again on page 197. It's a region of DNA made up of nucleotides that encodes a particular function. So the next question. This is 6 marks. It's a short response. You're going to be looking for multiple points here in your response. It asks for explain the role of helicase and DNA polymerase in the process of DNA replication. So you can refer back to that last slide where we spoke about this, and you should be able to go into a fair bit of depth here. Here is, pause the video, and I'll show you the answer in a moment. Here is the exemplar response for this question. You might have something like this, but notice that there's different ways to say the same thing here. So the role of helicase is to unwind DNA. You can refer to breaking those hydrogen bonds, and you can refer to creating a fork in the road here. In comparison, DNA polymerase works in pairs to copy one of the DNA molecule to produce 2 new DNA strands. You can talk about the 3' end and the 5' end. You can talk about the leading strand and the lagging strand. But remember, focus on just polymerase and helicase. That is all for today, guys, and I will leave you with a quote by this man by the name of Francis Collins, and he says, "The God of the Bible is also the God of the genome. He can be worshipped in the cathedral or in the laboratory. His creation is majestic, awesome, intricate, and beautiful." I love that. As we are talking about science, and as you guys are doing your textbook readings and answering your questions, how cool is that to think that, whoa, I am actually honoring God by doing this. So, hey, bless you guys. I hope that that was informative. I'll see you in class.